the center of the lobule. This vein we usually refer to it as the central vein, okay, of the lobule. So in the center of the lobule there will be a central vein. This vein will take blood all the way down to the inferior aspect of the lobule, into which we call it sublobular vein. Okay? The sublobular vein is a horizontal vein passing below the lobule, another one down here, another one, you see it here, and it will drain many central veins into it. Several sublobular veins will collect it into larger veins, we call them collecting veins. Then the collecting veins will form what? Hepatic veins. Two hepatic veins in the posterior part of the liver, which will drain into the IVC. Okay? So blood circulation, or the microcirculation of blood in the liver, starts with two structures, portal vein and hepatic artery in the portal triad, if you remember. I told you portal vein will bring 70% of the blood to the liver. This is related to the function of filtration, while hepatic artery will carry out 30% of the blood. This is related to what? To oxygenation. If you look within the lobule now at the periphery, you can see the portal triad here. This is the hepatic artery, this is the portal vein. Both together will drain their blood into a large capillary beds. Those large capillaries, we call them the sinusoids. Sinusoids, which means like what? Sinus, pocket. So because they are very large. Okay? Those large blood capillaries here, the blood will mix from hepatic and portal vein in these sinusoids. And it will go all the way until the center of the lobule, where we will have now central vein. So the microcirculation starts with portal vein and hepatic artery, passing through or across the sinusoids into the central vein. From the central veins, you will have what? Sublobular veins, collecting veins, then the hepatic veins, which will drain to the inferior. Okay? Now, the third structure, which I told you just now about it, the portal triad. The portal triad is at the periphery of the lobule and usually contains three structures. Okay? Those are branches from portal vein, hepatic artery, and the bile. Okay? So we have the hepatic laminae, we have central vein, we have portal triad at the periphery. What else? We have the blood capillaries. I told you we have large blood capillaries called sinusoids, and you see them extending from the periphery until where they empty here in the central vein. Okay? Those are the sinusoids, which are the spaces between the hepatic laminae. Okay? Those are blood capillaries. When I say blood capillaries, that means they have an endothelial line. Yes? So the endothelium that's covering the tunica intima, if you heard about it, in the hepatic artery and in the portal vein will extend into these blood capillaries together to join to become the endothelium of the blood capillary, the sinusoids. This endothelium will extend all the way until it becomes tunica intima of what? Of the central vein. Clear? So those are blood capillaries with endothelial linings. Those are the spaces you see here, those yellow spaces, okay? Which are, don't think here it's not continuous. It's continuous but at a lower level, as you see here. Okay? So if you look in a vertical view, you can see them here continuous. Okay? Some of them stand, then go down, then again like this. Okay? So those are the sinusoids, which contains blood from two sources. The larger source is the portal vein branch, and the other one is hepatic artery. So blood will mix within the sinusoids. Okay? And as the blood passes here, it will be filtered by specific macrophages called Kupffer cells. Okay? Those macrophages called Kupffer cells, and also as it passes, it will send oxygen to what? And the nutrition to the hepatic sites in the hepatic laminae. Okay? So as the blood passes, it will be filtered. You see these yellow cells, stellate shape, like stars. Okay? Those are the Kupffer cells, referred to Karl Kupffer, a German pathologist who described them for the first time. So Kupffer or stellate cells are phagocytes. Okay? Their function is the filtration of the blood. So now what we know? We know we have the hepatic laminae. What else? We have a central vein, we have a portal triad, we have the sinusoids. Number 5, we have macrophages. Living within these sinusoids, those are called Kupffer cells. The last thing we have to look at is the bile canaliculi. You can see green one up there. Those bile canaliculi are located within the hepatic laminae, in between the layers of hepatocytes. Okay? So if you look, this is a hepatic lamina made of 2 layers of cells. Between the 2 layers of cells, you will see here what? A small canal. So we call it canaliculi, smaller canal. Bile canaliculi will take bile from hepatocytes and drain it where? To the bile duct in the portal triad. Okay? So those are small ducts between hepatocytes that collect bile produced by what? Hepatocytes. Now this is a view, okay, a histological view of the liver. If you look carefully, you can see very flimsy or very thin connective tissue. Very hard to distinguish. But if you follow the arrow here, you can distinguish the hexagonal shaped portal or hepatic lobule. Okay? So this area here is the hepatic lobule. The area in the middle here is what? Central vein. You can see here what? Portal triad. Okay, another portal triad. Usually we have another one here. Usually we have 3 portal triads for each lobule. That located at equal distance. Okay? Usually we have 3 portal triads. You can see these pink layers, those are the hepatic laminae built of hepatocytes. And the white spaces here in between sinusoids. Okay? So is it clear for you? If you look at a section like that to distinguish its features.
Okay.
Now this is a greater magnification for the portal triad. See the difference in size. The largest structure here, where you can see some RBCs, is this is the portal vein. Okay? If you look carefully here, this is the hepatic artery. Okay? And you can see the elastic laminae in yellow here, the elastine. Now this smallest symbiocuboidal layer, okay, duct, it's all made of symbiocuboidal cells. Okay, this is the bile. Yes. So very large portal vein, mid-sized hepatic artery. The smallest is the bile. Concepts of liver lobules now. So when the anatomists and histologists studied the liver, they found out that it's built up, as I told you, structurally from the hepatic lobule, the hexagonal classical shape. And they call it as what? The classical or the hepatic lobule. Okay? So this is the first to be found. It's the first to be defined. It's usually explaining the function of blood flow or filtration in the liver, where the blood will flow from the portal triad, from the periphery across sinusoids into the central vein. Okay? Now later on, physiologists start to argue with this because they say that the liver contains many functions, mainly 3 functions. We said in the previous lecture in the gross anatomy. We said that first, filtration of the blood. The second is bile production. The third is metabolism, yes, of the drugs and detoxification. So there are 3 main functions. However, the classical lobule, which is the structural unit present there, explains only the process of what? Which function? The blood filtration. But if we look to the process of bile production, because bile secretion and production is go in an inverted way, okay, in an opposite way to the flow of the blood. So this lobule does not, as you see here, does not explain the process of bile production. So physiologists suggest that another imaginary, okay, functional unit, okay, but it's not structural, okay. So it's imaginary lobule. They call it the portal lobule. This lobule is explaining the function of bile production. How bile is produced? Bile is produced, as I show you, by hepatocytes that read the bile into bile canaliculi. From the center, it will be draining all the way into where? The portal triad. So the center for this lobule should be what? Should be the portal triad, specifically the bile duct, which collect the bile from surrounding classical lobules, okay, in the hepatocytes. So they agreed that a center would be the bile duct, the portal triad, and the periphery would be 3 lines joining the adjacent central veins. Okay? So the bile now can be explained, draining from the central vein into where? The portal triad or the bile duct. Okay? So this lobule, they call it portal lobule. Why? Because its center now is the portal triad. Okay? So So portal lobule explaining the function of bile production and secretion. Okay? It's imaginary, remember. Okay? You have to look and imagine that lobule there. Okay? It's not structural. The only structural lobule is the classical one, the hexagonal shape. So the triangular area, whose center is the portal triad, as you see here, where the bile will be collected, and the periphery is an imaginary lines connecting three surrounding central veins. Okay? By this, the bile will be produced from the center of each of those lobules into the bile duct. Now, after that, pharmacists concerned with what mainly? Drug metabolism, okay, and detoxification. Now, in this process, it depends on the survival rate, okay, of the cells, of the hepatocytes, and how strong they are to destroy these drugs or toxins. Okay, so pharmacists suggested another lobule related to the function of metabolism, okay, and detoxification of the drugs. They suggested that, they said, survival of the cells depends on the amount of oxygen reaching them. So it depends on oxygenation, okay, of the hepatocytes. More oxygenation, more ability of the cell to live and to destroy or metabolize the drugs, okay? So by this, they suggested that where the blood starts to flow, okay, from something we call it distributing arterioles. So if you look into a hexagonal, this is the one you saw in the lab. I added to explain it for you all. So when you look here, you will see that as the hepatic artery is passing in the portal triad, it will send an outstretched arteries, smaller arteries, we call them arterioles. And we call them distributing arterioles. Those are the distributing arterioles, as you see. And they go to meet another distributing arteriole from adjacent hepatic artery. So if you look in a cross section here, this is one of them. This here is a hepatic artery sending its distributing arteriole. This is another one sending another distributing arteriole. So those are the distributing arterioles from two hepatic arteries. As the arterioles is passing there, they will start to send the blood into the sinusoids within the lobule. Okay? So blood will start to flow from where? Into the central veins. Okay? As the blood flow, okay, the first area at the periphery here, as you see, closer to the distributing arterioles, will be oversaturated with oxygen. So the cells here will have more than enough what they want, oxygenation. So they will be the most able to carry metabolism function, okay, and detoxification. So this is the first area, which is a very strong here and a higher survival rate. The cells here have higher survival rate. So they call it zone 1, okay, or the periportal zone. Why periportal? Because peri around the portal triad, okay? So this is the periportal area or zone 1. Next, when the blood will pass here into the middle of each lobule, of classical lobules as we here and here, those cells will have just enough oxygen for themselves, okay? So this will be a little bit weaker than the first zone. They call it zone 2 or mid-lobular, in the middle of the lobule, mid-lobular zone. Now, the poorest area with oxygen supply will be where? The most peripheral, closer to the central vein. So not enough oxygen supply to these cells. So those cells will be the weakest and will be the first to degenerate, okay, under toxin effects, okay? So those cells now, they are located in which we call it zone 3 or centrilobular. Centri because they are closer to what? To the central vein. So we have three areas from where the blood flow for oxygenation from the periphery into the central vein. Those called zone 1, periportal, zone 2, mid-lobular, zone 3, the weakest, centrilobular. Okay? Three zones. And if you look to these zones, how the blood flow from them into the central vein, they all form what we call it diamond-shaped lobule. This diamond shape, okay, they decide to call it the acinus. Okay? Okay, so they call it the acinus. So whether you will hear the term portal acinus or hepatic acinus, this is indicating for the function of metabolism and detoxification. Okay? So this is related to that. So it's a diamond shape, as you see here in red, a diamond shape lobule with its center being the distributing arterioles. Those are outstretched branches, this is the definition, outstretched branches from hepatic arteries, okay, in the portal triad. In other words, it will be the line that joining two portal triads together, okay? The distributing arterioles, its periphery will be the two central veins, okay? So it describes the order in which hepatocytes degenerate, die, okay? In what? Due to toxic or hypoxic injury. What's hypoxic? Loss of oxygen supply. So how they die? Again, from the central vein back into the periphery, the distributing arterioles. Okay? So zone 3, then zone 2, then zone 1. Zone 1. So we have into 3 zones. Zone 1, the periportal. Zone 3, the centrilobular, and zone 2. Okay? So this is the hepatic acids. Now how to distinguish between them? Classical lobule, okay, or if you hear the word hepatic lobule or classical lobule is the hexagonal shaped lobule. This is both structural, you see it, extension, and functional, okay, unit. Okay? The others are only functional, imaginary lobules. If you explain the portal or the flow of bile, we would call it portal lobule. Okay? Portal, remember, the word portal is the key because it indicates the center of this lobule, portal triad. If you heard the word acinus, that will be the key for what? For the hepatic acinus or portal acinus. Okay? So 3 words would be your key words. Classical, the hexagonal. Acinus, the metabolism and detoxification of drugs. Portal, the bile production. 3 different lobules. Now, when we speak about the hepatic sinusoids and hepatocytes, the hepatocytes, as we told you, as you look here, they are arranged in what? In hepatic laminae. Okay? Those are arranged in plates and they radiate from the central vein area, as you see, into the portal triad or the periphery. Okay? Now, there are spaces between these plates, okay, and the sinusoids. These spaces, the spaces between them, the blood capillaries are the sinusoids. Okay? I told you that the sinusoids are nothing more than what? Blood capillaries. Okay? And those blood capillaries, they take the endothelial lining from tunica intima in the portal vein and in the hepatic artery and extend where? Into the central vein. So if you look here, this is tunica intima in the portal vein. You see? It continues here to become what? The endothelial lining of the capillary. Those blue squamous cells. You see them here? Okay? Then these cells will get to become what? Tunica intima of the central vein. Okay? As you see them here. So those are flat squamous cells. Those cells in the sinusoids are nothing than the endothelial lining of sinusoids, the blood capillaries. Okay? What they refer to them in the case of the liver, they call them sinusoidal lining cells. Okay? What we mean by sinusoidal lining cells are the flat squamous endothelial lining of the blood capillaries. Okay? Once we have these cells there, lining, there will be a small space. Okay? Can only seen under electron microscope. Okay? This space will be located between the endothelial lining cell, as you see here, and the hepatocyte. Okay? Clear until now? So in this electron micrograph, all of these flat cell here, you see it? This is an endothelial lining cell. Then you have the sinusoid. Within the sinusoid, we can see a very large macrophage, which we call it Kupffer cell. Okay? Now, after the endothelium, there is a tiny space from here to here. You see this area? This is called the sinusoidal space, or they call it space of Dizzy. Now, okay, referring to Joseph Dizzy, okay, a German histologist who described it. So they call it the space of Dizzy. After the space of Dizzy, you will have the large hepatocytes. Okay? So what are the sinusoidal lining cells? Those are the endothelial cells lining what? The sinusoids, the blood capillaries. So they are endothelial cells of the capillary. Those cells, they have fenestrae in them. Sieve-like. Okay? Fenestrae. Sieve, شو السيف؟ المنخل. Okay? Also, as they go in a flat with longer processes, they have spaces between each other, between the end of one and the start of another endothelial cell. Okay? So they have gaps there. This gap, the width of the length of this gap is about 0.5 micrometer. Okay? This indicates that if there is any structure in the sinusoid, in the blood of the sinusoid, it cannot pass into the sub-sinusoidal space, okay, or in the space of dizzy, unless it's less than 0.5 micrometer in diameter. Okay? So any particle or molecule less than 0.5 micrometer can pass below to become in direct contact with the hepatocytes. Okay? If it's larger, like the RBCs, very large, they will still stuck where? In the sinusoid, okay? Within the capillary. That's it. So if you look here, this is another electron micrograph. You can see the hepatocytes in the hepatic laminae, and those are the endothelial lining cells, sinusoidal cells. Okay? As you see here, this is another one, and they go, this space D, the space of dizzy, okay? Between the endothelial lining cells and the hepatocytes. And all of these black structures are RBCs of the blood, okay? So you can see the RBCs stuck in the sinusoid. They cannot escape into the space of dizzy, okay? The space of dizzy now, what you can see there, you can see many elevations from the hepatocytes. Those are called, like here, if you look carefully, these elevations. This is one, this one, this one. You see these elevations from the hepatocytes? Those are called microvilli. They are to increase the surface area in the space of dizzy, so they can increase the exchange of all molecules less than 0.5 micrometer. So the perisinusoidal space or space of dizzy is the space that located here, as you see, between the hepatocytes and the epithelial lining cells. So it will be this, all this space. D indicating the space of dizzy. So it's between the hepatocytes and sinusoidal lining cells. Okay? It's supported by reticular fibers. What's the reticular fibers?
Collagen type 3.
Collagen type 3. What's there in this space? There are plasma fluid, for sure. Okay? The plasma fluid of blood that can easily escape. Okay? Into this space. You can see RBCs are outside, they cannot get into the space, but you will see microvilli, as I told you, so many microvilli. All of these are microvilli. Okay? Those microvilli is coming from hepatocytes to what? To increase the surface area. Okay? So they're producing more efficient process of exchange. Okay? All of these are microvilli. There are some small cells. If you look to this one here, this is one of these, of the cells, very small cell. You see it? This is a cell here, and this is its nucleus. We cannot now determine what type of cell. It has to be one of two cells. Whether it's a fat storing cell there are specific cells to store fat there. Okay? These cells, we call them ITO cells, referring to the Japanese scientist who described them. The ITO cells, okay, a person called ITO. ITO cells are what they discovered. They are the area in the liver which store vitamin A. Okay? So vitamin A is stored there in ITO cells. Okay? And they have a very clinical significant feature. What's that? They are clinically significant because when there is injury to the hepatocytes there, okay, or to the parenchyma of the liver, ITO cells usually will become what? Activated. Once they are active, those are usually quiescent, okay, within the space of dizzy. They just filled of fatty droplets containing vitamin A. However, once there is injury, they will become active. Once they are activated, they will transform into what we call it myoepithelial, myofibroblasts. Myo indicated what? I told you the bigger structure? Muscle function. And fibroblast, fibrous secreting cell. So they will become active. In the fibroblast part, they will start secreting collagen fibers. So as they secrete collagen fibers, they will what? They will obliterate space of dizzy, preventing the exchange process, okay? And also extend these fibers into the sinusoids. In the myo part, they will have processes, okay? These processes will contain contractile proteins, similar to the smooth muscles. So they will start what? Squeezing, okay? Into what? Into the sinusoids itself here, all over, reducing the size of the sinusoid. So the processes will go all over the endothelial cells, okay? okay, and contract over them, reducing the size. So what will happen now? They will interfere with what? With the blood flow within the sinusoid from the portal triad into the central vein. Okay? So there will be obliteration or occlusion of blood flow, okay, and there will be what? Fibrous formation because of these, which called icosys. This case will be known as liver cirrhosis. Okay? So liver cirrhosis is mainly involved with what? With some problem where in the icosys. Okay, which will obstruct the blood circulation or microcirculation in the liver. Okay? So those are the fat absorbing cells. In addition, another type of cells there, which known as natural killer cells or bed cells. What's the function of these cells? They are similar to T lymphocytes. Okay? And some research suggests that natural killer cells and T lymphocytes share a common precursor. Okay? So those cells are immune cells. Okay? The natural killer cells, or they call them GIT cells. Domains now, if you look to the hepatocytes in their laminae here, if you look for example this, it has in this view only, but it has more if you look more. But if we look at this view, we will see that it has 4 surfaces. Okay? 2 surfaces adjacent to what? To another hepatocyte. And 2 surfaces are adjacent to what? To the space of this. Okay, and to the sinusoid. Okay? So if you look, what we mean by domain, the surface. So if we look to the domains of the hepatocytes, okay, we have 2 domains. Sinusoidal domain is the surface of the cell facing the sinusoid, or the space of the cell. The lateral domain, in janibi, is the surface that's facing another hepatocyte. Okay? So in the lateral domains, they will face other hepatocytes, okay? And this domain, you will see the function of bile production, okay? Where you see a small space area here, this is bile canaliculi, okay? Where the production of bile is going on. While in this area, you will see what? The space of Dissy, which is related with what? With the release of endocrine secretion, okay? And the transport of materials, exchange, taking oxygen, releasing CO2, for example. Okay? So in the lateral domain, you will see here what? The bile canaliculi. Within the bile canaliculi, you will see microvilli also projecting into the canaliculi. You see how the canaliculi is irregular in shape because of these microvilli. Okay? Why microvilli? Again, surface area. Wherever you find there is microvilli, like here in the hepatocytes or here, it's to increase what? Surface area. Surface area. So more production and secretion of bile. In the sinusoidals, the microvilli will project where? Here, this is the sinusoidal surface. And all of these are microvilli. What this space is called? Space of this. Space of this. Microvilli here increase the surface area 6 times. Okay? To facilitate the exchange of material and the secretion of endocrine products. Lastly, gallbladder. When we speak about gallbladder, okay, the 4 layers are no more clear. So what you will see here, I mean mucosa, submucosa, okay, muscularis externa, and the adventitia ulcerata. You will see instead just a layer of epithelial lining, okay, there, okay. This will be simple columnar epithelium, mubashara after it, a very tiny, thin layer of connective tissue. We would call it lamina propria. There is no muscularis mucosa. There is no submucosa. Directly after lamina propria, you will get into the smooth muscle, muscularis externa, or which we call it oblique fibers. Then you will have serosa and adventitia. Why you will have both? Because one surface of gallbladder downward is covered by peritoneum, while its upper surface is leaving on the liver bed. I told you in the bed of our gallbladder there is no peritoneum. The same in the opposite side of the gallbladder. So there, in the upper surface of gallbladder it will be adventitia. In the lower surface of gallbladder you will have serosa. And that's for the lecture.